In this video, we introduce a state-of-the-art diagnostic tool known as CARMEN, which stands for Combinatorial Arrayed Reactions for Multiplexed Evaluation of Nucleic Acids. CARMEN uses CRISPR technology to detect multiple pathogens in a single go. Results are given through fluorescent readout on a special microfluidic chip. With many diagnostics, there's usually a trade-off between high-throughput screening, which means testing lots of samples, and multiplexing, which means detecting many pathogens. CARMEN gives us the best of both worlds. CARMEN's power lies in its ability to detect multiple pathogens across multiple samples simultaneously. As a result, CARMEN provides highly sensitive, accurate diagnostics and enhances community surveillance for a wide array of pathogens. After watching this video, you will have a foundational understanding of the step-by-step process of CARMEN. Specifically, you will 1. Learn about how CARMEN works for the detection of multiple pathogens. 2. Understand the step-by-step process from sample collection to data readout and finally recognizing a positive result. And 3. Become familiar with key terms relevant to the technology. Scientists start by collecting patient samples to investigate for the detection of pathogens. Patient sample sources can include blood draws, nasal swabs, and urine collection, among others. Nucleic acids like DNA and RNA are then extracted and used as samples for CARMEN. The samples are loaded into a microplate and combined with primers specifically designed to bind to each pathogen of interest, such as SARS-CoV-2, an RNA virus that causes COVID-19. Primers for control sequences, such as RNase P, are also added. This human control gives us information about the extraction and efficiency of the amplification steps, independent of the presence of a pathogen. If these target sequences are present in the samples, the primers will bind, allowing nucleic acid amplification to proceed. Through RT-PCR, RNA is then converted into cDNA. Let's take a moment to go through an example using SARS-CoV-2. If SARS-CoV-2 is present in the sample, the primers will bind to the pathogen's target sequence. The primers are then used as a template to amplify or create millions of copies of cDNA for this region of interest. However, if the pathogen is not in the sample, the primers will not bind, so no copies are made. This process occurs concurrently with the primers for all the target pathogens being screened. Next, we prepare a sample master mix. This mix contains many essential chemicals for CARMEN to work, including 1. RNTPs, the building blocks of RNA. 2. FAM reporters. FAM is a green fluorescent dye used in molecular probes. Then, equal volumes of the master mix are partitioned into each well of the 96-well plate. Amplified cDNAs are individually added into their corresponding wells. The assay mix is made per pathogen by combining the pathogen-specific CRISPR RNAs, the assay controls, the T7 RNA polymerase, and the Cas13 enzyme. Now, the microfluidic chip is ready to be loaded. The assay master mixes are loaded into the assay inlets, and then the samples are loaded into the sample inlets of the microfluidic chip. These mixtures flow along designated microcircuits into the microwells of the reaction chambers to be combined. Up to 23 pathogens can be tested against up to 192 patient samples, although typically we run the assay for approximately 180 samples and 12 controls. We have now made it to the part of the assay in which the reactions start to happen. Each microwell now contains both the sample mix and assay mix for a specific pathogen. The Cas13 enzyme only acts on RNA, so it's time to make RNA. The T7 RNA polymerase enzyme drives the synthesis of RNA from cDNA using RNTPs as building blocks. CARMEN uses a CRISPR-based detection technology called Sherlock, which stands for Specific High Sensitivity Enzymatic Reporter Unlocking. If the assay mix droplet contains the CRISPR RNA for SARS-CoV-2 and the SARS-CoV-2 target RNA is present, the CRISPR RNA will bind to this sequence and activate Cas13. This is where the magic happens. The activated Cas13 protein acts like molecular scissors, cutting not only the RNA, but exhibiting collateral cleavage activity and cutting every RNA nearby, including the FAM reporters. When these reporters are cleaved, one end fluoresces. The more SARS-CoV-2 present in the patient sample, the more amplified cDNA, the more reporters cut, and the more fluorescence. These reactions occur in all the microwells at once. Interpreting the results requires the analysis of the fluorescence in every microwell using a heat map. By measuring the fluorescence for all our samples compared to a baseline value, we can determine the pathogen's presence or absence. We also run several controls to monitor for contamination or technical faults at different steps of the workflow. Let's recap. CARMEN is a tool that helps us detect emerging high-threat pathogens and supports our rapid outbreak response efforts. The technology undergoes 9 main steps. 1. Collection of patient sample. 2. Extraction of target from sample. 3. Amplification of target. 4. Preparation of sample mix with amplified target. 5. Preparation of assay mix with pathogen-specific CRISPR RNAs and Cas13. 6. Loading of the microchip with sample mix and assay mix. 7. Synthesis of RNA from cDNA and cleavage of targets and reporters. 8. Analysis of fluorescence and heat map generation. And 9. Interpretation of results. We hope that you enjoyed this video and find it helpful to learn about how CARMEN is used in pathogen surveillance. To learn more about outbreak response efforts, check out the rest of the videos in our series. Special thanks to our dedicated team of researchers and the Broad Institute video production team who supported the creation of this video.